Hi everyone, how are you today? So, after the Watson and Crick's announcement and reporting of this DNA, the accurate structure in model, okay, so everything is settled as a DNA as a genetic material was like uh, became a doctrine actually. Yes. So, but after such quite an accomplishment, Watson and Crick did not simply stay like, enjoying their lives as a with riding on their fame already. But on the other hand, actually, James Watson, until he finally died. Didn't he? I don't know. I'm not really quite. I didn't really check it. But anyway, he was really up until very recently. He was very active in as a chairman of the World Mecca, the the institute called Cold Spring Harbor was a world mecca in the molecular biology research in the east coast of the United States. And also he took the initiative of the famous Human Genome Project too. On the other hand, this Francis Crick, the photograph over here, also mainly spending the rest of his life on west coast of the United States also, and contributed and inspired so many important progress of the life science too. So here, this central dogma is one of his inspiration. That in reality, actually, what the central dogma is, what is this? This indication of the direction of the genetic information, stating that always the genetic information flows from DNA to RNA. What's going on? DNA to RNA and protein. Not more important thing is not the other way around. So the genetic information cannot go in reverse direction, starting from protein, going through RNA, and finally ending up in DNA. No, you cannot do that. You always have to go from DNA to RNA, and protein is what is called the central dogma, the flow of genetic information. So big deal. So what? I mean, I don't give anything about this central dogma thing, and everyone knows that already. Yeah, everyone knows that DNA is the center of the blueprint. What can you compare the DNA with? It's a blueprint of genetic information. It's the original, and RNA is a copy form. Well, once I compares the DNA as a cookbook, original cookbook, containing every recipes of the whole world dishes. And RNA is a copy of each particular individual page. On occasion, if you are interested in cooking, making pizza, and then you dig into the particular page describing several different pizzas, okay? And then you simply take a photograph out of the original holy place of where that the information is stored, which is nucleus. And then you bring whole bunch of copies. Usually when you generate photocopy, you don't simply make a single copy, but usually make multiples of copies. That's what you do when you do this transfer of genetic information from DNA to RNA. You usually make a lot of a single original copy. That's what we call as the process of gene expression, or in other words, we call this a transcription. And why do you make such copy? This is simply still up until this point, this whatever the form is still the conceptual thing, information only. It doesn't do anything. The instruction of how to make pizza cannot actually feed you. The food pizza, delicious, smelly pizza can actually fill your stomach, and that's protein. Okay? So there are some guys, there are some staff members who can understand this language, the instruction, the recipes of how to make pizzas, and then they grab each copy of this information, and then they can read, oh, okay, according to this instruction, the firstly, I have to bring this, and the second step, I have to bring this, and that's the process of this protein synthesis. And after finishing this, now you can, for the first time, you can see actual, tangible product of the gene, which is which can function. So anyway, this. So occasionally, not only does DNA provide the instruction for this protein making, but sometimes they do this thing, copying. At each round of cell division, they have to regenerate, so multiply this DNA replication also. So these are all such business of collectively we call as a central dogma. According to central dogma, always this information go this arrowhead directions. And these are just what I have described. So this DNA double strand, DNA double helix, this particular area is one part of a gene. Then this is a single copy of that region. So then at this point you may wonder, okay, DNA strand is double, so which of the two is actually the one being copied? Yes. You have a choice, like as an enzyme to make this copy, you can choose, okay, should I make a copy of this upper strand of the DNA or bottom strand of the DNA? Well, there is a certain rule, which we'll get to this later today of the lecture. So RNAs. So this is strictly information, and somehow, with some kind of magic and blessing, this information is directly converted into actual. So this ball-like, individual ball-like, a streptococcus-like, this thing, each ball stands for amino acid. So proteins are nothing but an assemblage, assembled form of individual amino acid. And when you have this, protein synthesis is finished. However, as a protein, the maturation is not finished yet. If you recall, some of the protein has to mature in terms of this the folding form, the correctly folded form. This is original primary protein just been out of the factory of the synthesis, if you call. And then they usually self-fold this into actual meaningful structure. Always a structure is the most important thing. Why the protein can provide so many different wonderful functions? Because of their unique ability to offer unique shapes. Remember, shape dictate function. Everything. Just like enzyme, the substrate catalysis or antibody, antigen interaction, and ligand receptor interaction, you name it. Everything is just a specific partnership, specific recognition, oh, you are special to me, and then binding, actual physical binding, and some differences are being made. That's the kind of a general paradigm of all these different activity of something happened in the cell. And by the way, the factory, this is the machinery, one single machinery that I referred a minute ago, referred as a staff, which is what? Right, ribosome. So this ribosome usually consists of these two parts, big, larger, small subunits, and it can recognize this messenger RNA and then read through in this direction. The working direction is actually the 5' end and 3' end of this particular messenger RNA to work through. And by working through, some assembly of amino acids can happen when we are going to look at it maybe next week. So, however, some creatures laugh at this central dogma. What's central dogma? Who cares about central dogma? Because dogma is, by definition, is something that you cannot violate, something like utmost principle. But not to them. These viruses, they don't care. They can go in opposite direction or reverse direction of that, the central dogma, what we used to know is totally useless now, then, if that is the case. So in other words, these guys, not all viruses, of course, some specific viruses, like for one notable example is, yeah, the retrovirus is, this HIV virus is one of the such virus called the retrovirus. It's not over here. Okay, so I will write down retro. These are group of viruses, including, so this HIV virus is one of these retroviruses. And what they can do that is, their genes are RNA. Oh, of course, many viruses have their genome, have RNA as their genome. So not a surprise. But what's unique about these retroviruses is they can start their whole this journey from this RNA. They can convert this RNA into temporarily into DNA. When they infect the host cell, these viruses, they have to put their genome temporarily in somewhere in between of the host chromosome. But host chromosomes are made of DNA and their genes are made of RNA. So they cannot put RNA directly into the host DNA. So what they have figured out is, okay, why don't we simply temporarily pretend that our genes are all made of DNA. So they convert their RNA into DNA because they have this particular, so this process is called the reverse transcription. And there is a specific enzyme allowing this conversion process. And the reasons that renowned Francis Crick formulate this so-called central dogma is human didn't have any idea about such enzyme is possible before. That is almost unthinkable. How can any enzyme convert RNA into DNA? We've seen so many different enzymes that can convert DNA into RNA, fine, but the reverse, the other way around, we have never seen. So that's the reason why they made this conclusion. But later, some molecular biologists discovered this enzyme. Wow! And actually, this enzyme called reverse transcriptase, we abbreviate simply as RT, reverse transcriptase, has been actually contributed, has been instrumental in the progress of all those modern molecular biology. We took advantage of their enzyme in a lot of different techniques. So anyway, this reverse thanks to this reverse transcriptase, certain viruses can go the opposite direction of what has been known as the central dogma. Is fine. Okay. But later, actually, more recently, it turns out that higher organism, including human actually also had this reverse transcriptase in our cell too. So it's no actually something totally unique feature, exotic unique feature in viruses only. Everyone can do that. We simply didn't know such a thing would be possible before. But now we know that. Uh, Even in some cases, in more recently, this conversion, starting from protein and making RNA out of the protein, has been demonstrated as also possible these days. So all this original order of central dogma has been screwed up in these days. It has only some historical value remained these days. Yes?
센트럴 도구마 예외 현상 가운데 그 광우병의 원인이 되는 프리온 잡질의 감염 과정 이런 예외도 또
I don't think so. That's a little bit different. The prion case, originally the prion people, some people thought prion was also a virus, but prion is simply protein, some weird protein. It has nothing to do with the DNA, genetic material. But those proteins are so unique or weird in that those proteins is highly resistant to those any harsh condition. They can be heat resistance and also acid resistance. So it can, through food, it can come to us and then infect our brain cells. And once they do that, they behave something like a zombie, so that convert normal their friends protein into their one of those such a prion protein, and because of that our brain cells are all disrupted because of it. So that's not that's irrelevant to the central dogma. So related with this central dogma, actual main question they would like to pursue, especially in this course, it would be something can be summarized as this. So this DNA, the DNA was generating information fine. We finally have settled. But what exactly the molecular nature of this DNA that can serve as genetic material? Still, we have a vague idea of, okay, DNA is a gene. It functions as a gene. The question is how? How this DNA can serve as some kind of information of life, genetic material. And the next how is this. Okay, DNA is a simply genetic information, then it has, according to this central dogma, it has to be transferred to temporarily to RNA. How? How can be done? And finally, then by using simply this RNA, how cells convert this information into actual protein is the step that we are going to follow for the remaining chapter content of this. So first, we have to officially define what exactly a gene is, right? We always talk about genes. Well, what is that actually? So, more narrow definition of a gene is something directly related with this protein. So you can say that more conventional, but I will call that as a narrow definition of a gene is some segment of DNA, not all DNA, but according to this, along with this chromosome, some portion, some area, some segment of the DNA on the chromosome, okay, that bears the information, meaningful information for protein synthesis. So like if I am allowed once again to compare this gene to this original that the whole recipe books. So if you turn up any particular page, some pages probably you may not be able to understand all this uh, gibberish nonsenses. So you cannot make any sense out of a single page. And such area is a useless area, for example, and those area on those content on that particular page cannot serve as a gene. But on the other hand, in some page, actually, if you read, oh, you can make yourself understood. Oh, I see. I see what you mean. So this is what something. If I follow this instruction, maybe somehow I can somehow try to make a list, jajangmyeon or kind of, then it is a gene, is something that you can say about. But broad definition, okay, so gene, DNA area that can give us some specific instruction for how to make protein, the specific information, coding information. Protein coding. Okay, what kind of amino acids you need to bring, in which position you need to bring this particular amino acid. You have 20 different choices in each position, for example. And that's what the job for the gene. Gene tells you exactly which order and what particular position, which particular amino acids will come. It's the classical definition of the gene. However, broader definition of gene is also more inclusive in that it expanded into not only this protein, but any segment of DNA that can, as long as this area can be converted into RNA, then it's technically, it is the part of the gene. It's more correct, actually, more accurate, and also broader definition of the gene, because not all RNAs can directly relate with. Not all RNAs can directly code for proteins. Some RNAs, if you read the RNA sequences, you have no idea about, you cannot get any information regarding what kind of a protein in terms of this amino acid. But even with that, these RNAs are extremely important for your survival. So how are you going to regard this type of RNA? So the area that is responsible for making such RNA also should be considered to be a legitimate gene. That's why this definition should be expanded to this. So simple reminder that chromosome, if this was one single cartoon of chromosome, if you untangle, so expand, so each naked DNA double helix level, only some area like this, certain regions serve as such genes, which means it can generate RNA copy. On the other hand, some other areas simply sit there doing nothing, it doesn't provide any RNA copy, it's simply there. Before, in older days, like in 1980s and early 90s, so scientists even used to call this area as a junk area. But that's just a totally inaccurate estimation, because even this area, even though they are not generating directly any RNA product, but still, in many different ways, provide very important supporting roles. More and more, they are realizing the relative importance of these non-coding areas too. So there are 6. Actually, depending on how you think, there are more than one. So there are more than 6, probably 7 different kinds of RNA, but usually it will be more appropriate if we simply settle. Okay, there are 6 different types of RNA existing cells, especially in eukaryotic cell. In this left panel, 3 different RNAs are more conventional, classical type of RNA. So probably, and in my days of as a graduate student, even I learned only these three different types of RNA because people didn't know even these three. So these three new RNAs arranged on the right panels are more recently discovered RNA. So their names are and their abbreviations are summarized as like this. I don't have to repeat them. So mRNA, messenger RNA, blah, blah, blah. Okay, so once you didn't have the question last lecture, you will also have this copy this time because I also have uploaded these questions in Snowboard too. So RNA, so she really misunderstood that RNA doesn't have a partner, or maybe thinking that DNA automatically should have partner. No, none of the cases. Nobody has there any kind of a designated partner. The only way that it looks DNA had partner while RNA doesn't is because DNA and RNA, regardless, the only reason why they can have this kind of relationship is because of what? The complementary base pair things. So how can RNA have this base pairing? Okay, so probably, so I gave up totally. I thought you already took this homework, which was the part of the take-home lecture. So if you listened or viewed that lecture, makeup lecture, probably I didn't have to put up this again. But anyway, I will just go over this thing again. This is a fundamental thing. I should have gone at least covered this before we enter this molecular biology stuff. So left side, this is a one single strand of a nucleic acid, whether at this point you don't know whether it is a DNA or RNA because the difference between DNA and RNA is made at the component level, chemical, so nucleotide level in the beginning. So this is a structure, chemical structure of a nucleotide, simplified, of course. There is one phosphate group and there is one sugar. And like I went over the earlier last lecture, there's one nitrogenous bases. Those are three part one component. Nucleotide consists of three different chemical parts: phosphate, sugar, and nitrogenous bases. And the thing is, in this sugar, there are two different alternative sugar can come. Everyone can quickly recognize, distinguish the difference between these two sugar, right? It's just a matter of time. The differences reside over here in this position. This sugar on this right side has an OH, and this left one doesn't have O on the same position. So this O, oxygen, because of that, the name, this is called the deoxyribose, the sugar, deoxy. Oxygen has been taken out, deoxy. And this is a regular sugar, ribose sugar. So this is actually the more original than this one. And this sugar is present in the RNA making nucleotide, whereas this sugar only appears in the DNA nucleotide. That's why the name DNA. The reason why we call DNA as a DNA is a deoxyribonucleic acid, DNA. And the reason why we call RNA as an RNA is it starts with ribonucleic acid, so DNA and RNA. So that's the first obvious difference between DNA and RNA. But another difference is over here. On this panel, 5 different choices to become alternative choices to become this position as a nitrogenous bases. You recognize 2 purines, right? 2 purines, and 3 pyrimidines. So when you make up DNA, only these 4 nucleotides assemble and make any one of those 4 different nucleotides. That's why adenine, guanine, cytosine, thymine, A, G, C, T. However, in the case of RNA, this thymine is switched with the uracil. Everything else stays the same, but only this thymine is gone in the case of RNA, and instead of this thymine, uracil take the part of the thymine. What do you mean take the part of this? You compare this thymine and uracil with your eyes. What is the exact difference? The only difference is on this position, the methyl group. And uracil and both uracil and thymine, the area where they actually use in forming that complementary base pairing is this region. So thymine and uracil have no differences, chemically speaking, in their ability and also specificity in generating, forming the complementary base pair. So exactly uracil can exactly replace the role of a thymine in the case of RNA. So uracil simply will be able to pair with A, and in RNA, this guanine, same guanine is there, will pair with cytosine. So in other words, RNA has no any handicap or disadvantage of establishing their respective complementary base pairing. Just like DNA, RNA can exist in 2 double helical RNA structure too. And what do you think? If I ask you, would DNA and RNA hybrid be possible? Yes or no? In nature, what I mean. In nature, would the DNA, and one strand is DNA and the other partner strand is RNA be possible? Yes or no? Yes, they do exist. They can do that. Nothing prevents from the complementary base pairing. Is a chemical reaction. As long as the particular like guanine nucleotide and cytosine nucleotide is there, they don't care about whether it comes from DNA or RNA, they will simply pair. And in the case of uracil, simply they also has no problem of pairing with adenine of DNA. So they will. So why, even with that, why RNA exists in single strand? It's a good question, isn't it? RNA can do this, but still RNA mostly in the cell, all those RNA exist as a single strand. They prefer. They don't want to be bothered with this, hey, should we have some kind of partnership like those DNA guys? Wouldn't it be nice? No, no thanks. Why? Because this very subtle difference of having one extra oxygen in the sugar make ton of differences. So it bothers when they try to have this double-stranded, it can only exist, stability, they can only stretch on a very short distance. DNA is how long a DNA molecule can go. It's huge lengths. But RNA, if you want to accomplish such long distance relationship of double-stranded of RNA, they immediately, hey, I don't like this because it's chemically doesn't fit because of this. So they tend to. But if you want to do that, RNA should, if they want to, if they are ordered to behave exactly like DNA, they have to maintain such a long double-stranded strand just like DNA. That's impossible for them. So they only have this temporarily, they can have this small area, short distance, the complementary base pairing based double-stranded things they form, and more preferably they tend to form their own like self intra-strand, self-complementary base pairing. And that is the reason why actually, as I explained earlier, there is a reason why RNA, unlike DNA, can behave as an enzyme, ribozyme, such thing. So we'll get to that. So briefly, scanning through these different types of RNA, I would like to simply emphasize and distinguish the concept of this coding RNA versus non-coding RNA. Messenger RNA is a coding RNA, obviously, because the sequence present on the messenger RNA, which is exactly copy of certain area, certain region of the DNA. What do I mean by copy? Copy means complementary copy. The reason why RNA can copy the DNA is because the chemically RNA nucleotide can temporarily have the complementary base pair with the DNA. So they follow some sequence in their complementary the fashion, and then they separate from the DNA region. That is the nature of this RNA. But anyway, this messenger RNA is the exact copy of the protein coding gene. That's why this RNA, the messenger RNA, can code the amino acid information. So those messenger RNA's information can now later can be converted into the building of a protein, because each segment of a messenger RNA specify what kind of amino acid to come. That is what I mean by the coding RNA. It bears the amino acid information. So if you simply follow the order of this messenger RNA, you figure out the order of amino acid of a particular protein. So you simply assemble each amino acid in such order, then voila, now this is exactly the protein that you have intended to make. So just like this. So originally DNA, this DNA region, template DNA. So it's just a matter of which of the two you are actually going to choose as a template. So in this occasion, this messenger RNA, which of the two, top or bottom, did this messenger RNA choose as a template? Top or bottom?
만약에 그거 여기 밑에 있는 서열을 템플릿을 한다면 어떻게 돼요?
So that's what I'm asking. That's my question. Which of the two, top or bottom?
Bottom.
Bottom was the template?
네.
Why?
이것을 바텀에 있는 걸 그 템플로 사용하면 어떻게 돼요? mRNA.
Then it should be, the sequence should be something like this. The RNA sequence. Since this RNA chose this sequence as a top sequence as a template, that's why the bottom DNA sequence is the sequence of this messenger RNA, right? So that means this RNA took this top sequence as their template, right? Get it? Okay. So now, then, once you become a messenger RNA, now you have only this messenger RNA has this kind of a concept of a codon. Only messenger RNA. No other RNA has this concept of codon. What is codon? 3 nucleotide. It's something like the word. Using alphabet with 4. Using 4 alphabet, how many different ways you can generate a word? That word, there is one condition. That word has to be 3-letter word. There's a concept of a codon. So each different combination of these 3 nucleotides can specify one particular amino acid information. So for example, okay, if this UAC, this particular order, U first, A and C, starting from 5 to 3' end, then this particular amino acid called tyrosine is the one. And UCA will code for serine, and GGU, glycine. So try not to memorize all these things. It's useless. There are 64 different combinations can come on. So anyway, each different combination of the 3 letters of 4 nucleotides can specify any kind of amino acid. But the problem is there are only 20 different amino acids. You have capacity of 64. Since you are using 4 letters and then picking up 3 regardless of this order, then now there are 64 different ways. So that will be explained later when we get to that. So anyway, that's the definition of a coding RNA. Because messenger RNA is designed, their job, designed to this job. Each particular sequence should match different type of, why? Because later, once this messenger RNA is generated, then somebody will interpret. That's why it is called the translation, this process. Somebody named ribosome will interpret this. I can read this thing. I know what this means. Okay, so that means bring tyrosine over here. So, hey, tyrosine, why did you come over here? And tyrosine will appear. And the next, UCA, ribosome, no. Oh, this means serine should come. Hey, serine, is there any serine? Then serine will come. And ribosome will connect. When they arrived together in proximity, then ribosome will connect this tyrosine and serine together through particular chemical bond we call the peptide bond. That's the name. That's how you go all the way till the end when you encounter with, okay, stop it. You will end here type of a signal will be encountered eventually. Ribosomal RNA is, on the other hand, first, this is non-coding RNA. Remember, messenger RNA is the only coding RNA in the cell. Everything else is non-coding. So, ribosomal RNA is, the reason why it is called the ribosomal RNA is that it's there, it's present in the ribosome. So ribosome is simply a mixture of several kinds of RNAs and the several whole huge, something 30 or 40 different proteins mixture. That's ribosome. So without ribosomal RNA, ribosomes cannot be assembled. So it is extremely important. And also, more importantly, actually this is the one particular ribosomal RNA is the one responsible for connecting different amino acids. So it is actually acting as an important enzyme. It's a ribozyme. And another extra gravy is look at this. Where is the 5' end of this RNA, ribosomal RNA? I cannot even find. Oh, here. There you go. This is the 5' of this RNA. And where is the 3' end of RNA? Here. If you unfold this RNA and then stretch it out in linear fashion, it will be extremely, extremely long RNA. But remember, what did I tell you about the property of an RNA? They tend to form self-complementary base pairing. So here, this is the region. They are actually having this complementary base. This is the region. They are forming their complementary base pair within itself. And there is a reason why they ended up this particular shape in 2D. If you view this particular shape in 3D, the shape would be much more dramatic, wouldn't it? Right. So RNAs is really good for this type of thing. They can form this internal self-complementary base pairing rather than try to pair with any other partnering RNA or DNA, unlike DNA double helix. That's the nature of RNA. Anyway, it goes to the ribosome. Right? tRNA is also non-coding RNA, and its job is to work as a carrier of different amino acid. Bring amino acid to the site of a protein synthesis, which is the ribosome. tRNA also, starting point, the 5' end of tRNA is over here. 3' end of the tRNA is over here, but they don't, rather than they exist in linear, stretched out form, tRNA, as this is the area where they have this complementary base pair, and this is 2, and this is 2, this is 2. Only those regions having this loop, why they have this loop? Because in this area, they cannot have this complementary base pair because they don't have their partnering complementary bases exist. Such area will remain as a loop. But other areas which they can find, if this position, for example, if this position was an A, then it will have a complementary base pair with this T, not T, U, because it's an RNA. Right? So they are having their self-complementary base pairing as an RNA. And as I mentioned, that since this is a 2D, two-dimensional view, actual three-dimensional view will be like this, more specific and dramatic, like this. Wobble.
RNA 전사와 번역에 어떠한 도움을 줄지,
We'll get to that later. Okay.
유전자 발현에 어떤 도움을
It has nothing to do with the gene expression, but this position is very important area. I will cover this later because this is something to do with actual protein synthesis process. Okay, so be a little bit more patient, and I will get to that later. Not today, but already it's almost 3:15. When I'm going to have a time for question and answering again, I'm worried. So tRNA has something is, since there are 20 different amino acids at least in the cell, 20 different amino acids participate in the protein synthesis. So in other words, if tRNA's job is to work as a carrier of different amino acids, at least the minimum there has to be 20 different transfer RNAs present. Each RNA, so the very 3' end of this RNA is actually the area where this amino acid is attached to this point. So each tRNA can come to the ribosome carrying this particular amino acid to the site. So there are a minimum 20 different, but in fact there are 60, more than 60 different kinds of transfer RNAs exist in the cell. Okay. I will explain that later, not today. But snRNA is more weird RNA, which stands for small nuclear RNA, exists in the nucleus only. It is also non-coding, and there is some mysterious segment called introns in the eukaryotic messenger RNA. Useless intervening segment of the RNA, which you don't get to see in the final product of a messenger RNA, but it's still there in the first place. And then during the trimming procedure, during the processing of this messenger RNA, they are removed. We are going to take a look at that introns thing later. But anyway, those intron, the segment of the original messenger RNA, have to be removed. And that job is done by this particular RNA. And SNO, SNO RNA is doing the similar thing for the ribosomal RNA. Ribosomal RNA is also being clipped to become the final version of the mature trimmed ribosomal RNA, just right after the synthesis. So they are some very specific, their particular function is something that suffice to remember that for you. You don't have to know too much details about this type of RNA. The microRNA, miRNA is called the microRNA, is more interesting, a little bit more interesting in that regard among those three new types of RNA. It is also non-coding RNA. However, this non-coding RNA is very deceptive non-coding RNA. There's almost mistakenly you can consider this as an actual messenger RNA. But when you try to follow actual sequence, what kind of amino acid is there, you will soon be disappointed that, oh, this is not going to generate any meaningful protein, because it's like deceiving you as if it is really, because it is extremely similar. Its sequence is very similar to known messenger RNA, but not exactly identical. And its role is to regulate this particular target RNA's gene expression. Okay, when I said that, probably a lot of you, what? I don't get it. Okay, that's it. Only that far. I will. So this microRNA is the one regulatory RNA, which is microRNA is not actually giving you any real information of the protein synthesis, but it is extremely similar to real existing messenger RNA, and by virtue of their sequence similarity, they can somehow control actually the production of this particular messenger RNA that is very similar to its particular microRNA sequence, is their job. So it's such a really wonderful new discovery, and it's very fancy its job is. So it's very hot research area, one of those very hot research area in this modern molecular biology. And luckily, and also proudly so, and she is a professor at Seoul National University, and she is the leading expert and world expert. So some actually predict that she will be the one of the first who receives the Nobel Prize in this area as a Korean. And she is leading this area of this microRNA research in worldwide. And this figure describes how this microRNA controlled this expression of gene is the figure, but probably a lot of you didn't get it, so never mind. So anyway, this area, sequence similar area, so they can have this RNA interaction. So this is an area where you can see actually in the cell, RNA double-stranded interaction is being made. And because of that, this RNA will be destroyed so that the expression of the RNA is suppressed due to this. That's what I'm saying. this microRNA can regulate, control the gene expression. And more interesting story, I can go more and more about this microRNA, but simply I will finish this story of microRNA saying that actually this was evolved through the mechanism against, the defense mechanism against infecting viruses. Viruses are coming into our cell as a single-stranded RNA, mostly. A lot of RNA comes in the form of RNA outside from us. So in order to prevent, detect those foreign RNA and then destroy them, host cells have developed this type of mechanism. So microRNA was one of them, and later it was converted into the one that actually controlling its own gene expression too. But anyway, so gene expression. Okay, this gene expression is a type of a little new thing, so I will just stop over here and then make to your try to cover a portion of your questions from now until the end of the class today. So gene expression thing I will cover the next week. And still, some of the students, a lot of you are hopefully now get them, but still, some of you have a little bit of this confusion, so I will go over this once again. Why? What's the exact definition of the dominant and recessive trait? It's nothing to do with any superior or strength of the gene themselves, but simply it's just a matter of whether it can be observed or not, the effect. Whether you can feel, observe, or recognize any the effect of the gene's function, then it should be dominant. And recessive trait is the reason why it is recessive is when they are present in the status of this mixture, like heterozygosity. When the two recessive trait, the same recessive trait exists as a homozygous status, obviously that recessive trait will show. However, as long as one recessive trait and one dominant trait exist as a heterozygous status, then automatically this recessive trait will be hidden, will not be shown. Because this dominant trait will cover up. Dominant trait will take over and you shut up, I will now be in control. That's the definition of the dominant and recessive trait. Okay. So the principle, we already know exactly the mechanism why a certain trait is become dominant and why certain trait become recessive. We already know that. Simply, it's just a matter of explaining and understanding. Because of that, even without testing actual the effect, as long as you can guess the actual function of this particular gene, then you can predict correctly that whether this type of alteration, this mutation will become dominant or this mutation will be recessive. That's the beauty of the molecular biology. Like, let me give you another example, like in the car. In driving a car, let's say you have two gas pedals. In a normal car, you have one gas pedal and one brake pedal. But let's assume that since you are a diploid organism, let's assume that also in your car, you have two gas pedal and two brake pedal. Now, while you're driving, these can be broken, can be out of order. Let's consider this, the malfunction of the gas pedal first. There are a lot of different ways of gas pedal can be broken. However, one particular way of this gas pedal be out of order is usually sometimes this gas pedal can be flawed, down, doesn't pop up again. You push the gas pedal, then as soon as you remove your foot, then this gas pedal will be pop up again. That's the mechanism of the gas pedal. Everybody drives, right? Everybody has your own car, so you can drive. You don't? Oh, okay. So anyway, that's the job of the functional mechanics of this gas pedal. But sometimes actually it's a real the malfunction happening. And this pedal, you push this gas pedal and then go down and doesn't pop up. So it stays there as an activated form. This is one kind of such malfunction of a gas pedal. Now, you have two gas pedals. If one of these gas pedal has this flawed malfunction, would this become dominant or recessive? This flawed gas pedal will act as a dominant or recessive, tell me. So that's the key, that's the clue how you determine whether it's going to be a dominant or a recessive. You have two gas pedals, and one of them has this malfunction. Oops, it doesn't return again. So the car will just go like 급발진. So if you have brake, you can stop this car by stepping on the brake. But anyway, let's forget about this brake. But as far as this brake, I mean the gas pedals function concerned, would this malfunction will behave as a dominant or recessive? It's going to behave as dominant. What does it then mean? As long as, even though the other copy, the other pedal working normally, but it doesn't help. Just because this pedal is still functioning, that doesn't stop this malfunction of this flawed gas pedal. Still car will go. Even this pedal, another copy, extra gas pedal stays the same, normal, acting normally. So this type of malfunction will behave as dominant. Can everyone agree? Probably. None of you seem to agree to my. So that's this type of new function. You have this new function. The normal gas pedal, you push and then car goes, and then you step out of this gas pedal, then car stops. This function is a normal, so to speak, normal gas pedal's acceleration function. But among many different ways of having this out of order the gas pedal, this particular flawed version of the malfunction, once you have this, normal gas pedal cannot have any control over this car. That's exactly what's happening in the dominant mutation. On the other hand, if you simply have one gas pedal is not working at all, big deal. You have another gas pedal still working. So, oh, this gas pedal doesn't work. I just push this gas pedal, but still car doesn't go. Big deal. I have another gas pedal. I will just push this gas pedal. So in this occasion, this gas pedal's malfunction is recessive. Try to think of brake malfunction. Probably it will become much more clear. Brake. Let's say you have malfunction in the brake. So you press, no matter how hard you press the brake pedal, car doesn't stop. That's the malfunction. But luckily, if you have another brake pedal working normally, then one malfunction of this kind of brake pedal, there's no problem. You can still stop the car using the normal, original function of a brake pedal. So in this malfunction of the brake pedal, it's going to act as recessive, as long as you have another normally working brake pedal is there. That's the difference between dominant and recessive definition. That's all I can try to explain. So, like this, it is nothing to the majority. Although, if one particular mutation is something lethal, that means because of this mutation, it will kill you. Even though if that mutation is recessive, but there are chances that these two mutations come together as a homozygous, two recessive, an individual having this lethal mutation will be, as an homozygous, will be eliminated out of the population. So the more you live and the more generation you go through as a population, then this lethal allele will be eliminated of the population. So later, what you count, when you count those, the ratio, and this dominant allele will be present as a majority of them, because by chance, it's a chance event. There are 4 out of 1, when you have this kind of a mating, 4 out of 1 in terms of the ratio, you will have a chance of having this lethal mutation together as an homozygous. Then you are lucky. You are unlucky, so that you have to be out of this the cellular business of living. So if you keep doing this as a population, then this dominant allele will become the majority. On that particular special occasion, yes, that's the case. But if this recessive or dominant the trait does not affect too much of your viability, and the popularity is nothing to do with their like dominance or recessiveness. That is also the thing that I hope you will be able to understand. And yes, this as a gene, being able to transmit itself to the next generation is so another, maybe you can call this as an important function, but as far as this actual function of the gene, this genetic transmission is nothing to do. This is simply a mechanical thing. So actual gene function is nothing to do with. So functionally, and the other thing is, I didn't say this as a recessive trait as a functionally useless. No, actually the wording that I had was this recessive gene lost its original function, unable to exercise its original function, its absence. Instead of labeling them as a useless, but simply I had something to show for you, but somehow because of the mutation, I lost that one. So unfortunately, at this moment, I cannot show this to you, especially when I am present with the other the original. Now it became dominant, original status is the thing. This is what I call as what? So all those previous examples of the different traits are the result of a mutation. Means mutations are by definition the changes in the DNA sequences. However, sometimes even without having this DNA sequence alteration, you can have sometimes the changed trait. That's what we call this epigenetics, as opposed to the classical Mendelian genetics. And this epigenetics is the hot topic of this modern genetics nowadays, because there's something, at first it looked so weird and it looked so strange and also fantastic. That's why. And it's nothing to do, actually the reason for this epigenetics is expression. By having different expression level, you can also achieve the different traits, and that is all those results. And some of these epigenetic traits can be influenced by environment. Environment can heavily influence this different epigenetic status. But now the question is, this epigenetic status influenced by environment, can this be also transferred to the next generation? Sometimes yes, even. It's very strange. That's the mystery. Sometimes it is yes. Sometimes something that is influenced by environment so that you change your expression level. You have the same gene, but simply showing the differences in terms of how much RNA you are going to make. Some individual make small amount of RNA of the particular gene. Some other individual make much larger amount of RNA of the same gene. And a lot of times that can be the result of a different environment, actually. When some individual was subject under the different environmental condition, that change will usually occur. So if you compare the DNA sequence of these two genes, there is no difference in DNA sequence. But still, you have a difference in the amount of RNA making. It's because of the outside environmental input. Now the question is, just like you stimulate your muscle and after going to the gym, and then you develop your muscle, but everyone knows that this developed muscle is not going to be transmitted to your sons and daughters, but sometimes it does. Sometimes some of those traits that you have developed in response to the environment mysteriously copied to your next generation, and they show the exact same response under such condition. So that's one of the mysterious the aspect of the epigenetics. What I'm saying is not all those epigenetic traits do this, the heritability transmitted to next generation, but at least some of them do that. So that's very funny. Okay, one last question. What do you think? Which one of these two actually was the original and the other one was the mutant version? The one having the capsule, the one not having the capsule, the gene, the same gene, but mutation occurred, and so these two alternatives, S form versus R form, S form still generates the capsule, R form cannot generate the capsule anymore. Which of the two was done actually the result of the mutation? And the one was the original. What do you think? I would guess this one was the original and this one was the mutant form. It's my guess. Why? Because you have something already functional. Which one is much easier? The one not having. If you think the other way around, in the first place, you did not have any ability. You cannot do anything. You cannot produce any capsule. But all of a sudden, overnight, somehow you became something that can cover up entire those outside as a capsule. It's highly more unlikely. Then you used to have this capsule, but something happened. And because of that, a small malfunction, now you're no longer able to make the correct form of a capsule, so you lost that ability. It's a more natural way of, if you think about the way the evolution goes through is. Of course, it cannot be applied all the universal cases, but that is the usual way of speculation. It is much easier to lose your ability once you have, rather than reinvent, totally inventing a total new function. That's much, much more difficult. Okay, goodbye and have a nice weekend. It's only Wednesday, but